This is an instructional video for the Prokaryotes lab. I wanted to show you from our Moodle page. This is our Bio 112 section NT1 Moodle page. And if you look on the left, if we want to do any assignments in the Prokaryotes section or on the Prokaryotes page, we'll click on that and you can see that these are the slides. So you can look through the slides to kind of help you organize the content. And then you'll need to do the study plan. It was due on June the 10th. If you haven't done it, I have extended that due date to June the 15th, just because it was a little over half the class that did not get the prokaryote assignments in. So I am extending the date. I think most of you did the homework and the quiz. So those are actually due yesterday, but I'm extending the date until June the 15th, which is Tuesday of next week. Now, the lab, the assignment Lumen Prokaryote Lab, says complete the lab report above on your device or else print out the lab report and answer the questions by hand. So again, I'm extending this due date until June the 15th because as you can see, 7 of you submitted the lab, but 6 of you didn't. So that's a little bit less than half of the class. And I did get some emails from students who had questions and didn't understand some of the things in the lab. So I'm going to show you. First, you'll click on the lab report. This is what I expect for you to fill in and turn in. And then you just read through it. All right. It gives you the objectives of the lab. And then the procedure, it says do the reading prokaryotes, and that includes this introduction, so you'll read through that. The shape of prokaryotes, and then a section on cyanobacteria. And then number 2 is a little video that you'll watch on aseptic technique. While you watch that video, you'll answer these questions. I don't believe students had trouble with that. I've graded this lab, and for the students who completed the lab, it seemed like they did fine on question number 1 and question number 2. But when you get to question number 2, it tells you that you can find the prepared slides of typical bacteria in the additional resources folder on your Moodle page. So in order to get the information so that you can draw a picture of the coccus shape, the bacillus shape, the spirillum shape, you're going to need to, I'm going to just minimize this for a minute. You're going to need to go back to that back to that Moodle page and click on additional resources for the Lumen Microbiology Lab. And for the first part, you're going to click on prepared slides of typical bacteria. And there should be 3. Yep, this one is shows you what Staphylococcus epidermidis looks like under a microscope. Once you have performed a gram stain on it, it's purple, so that means it is gram positive. The bacteria are spherical or coccus shaped, and they're growing in clusters. And these are Escherichia coli, or more commonly known as E. coli bacteria. They have stained pink, so they are gram-negative bacteria. And they have a rod shape, or an oval shape, and that's called bacillus-shaped. And then we have Spirillum volutans, which is staining pink, so it's gram-negative, and it's spiral-shaped. And another example of, these are just gram-positive spiral-shaped bacteria. Okay, and so then we'll go back to our lab. And scroll down. All right, then for the prepared slides of cyanobacteria, those are also found in the additional resources folder. So we'll go back to the additional resources folder and click on prepared slides of cyanobacteria. And there's going to be questions on the different types of cyanobacteria. This one is called Anabina, and it grows in long filaments. There's a question about the function of a heterocyst. Most of the cells that are connected to form the filament are vegetative cells. So these cells along here are vegetative cells. And vegetative cells are just the ones that divide. Most of the cells that produce the Anabina are vegetative cells. They're the cells that help the Anabina filaments grow longer in length. And the heterocyst is, you could look this up online to find the answer. Some of you were wondering how to find the function of the heterocyst. You could just Google heterocyst, and I've read the Wikipedia, and it's the Wikipedia explanation is just fine. So heterocyst is a cell that is capable of converting the nitrogen gas in the atmosphere into usable nitrates and nitrites for the Anabina bacteria. So what it does is, the process is called nitrogen fixing. So it fixes the nitrogen in the atmosphere into a usable form of nitrogen that the Anabina can use to produce proteins and nucleic acids. So living things can't just take nitrogen from the atmosphere and use it. Nitrogen gas in the atmosphere is not usable by living things. But living things need nitrogen. They need a source of nitrogen because our proteins and our nucleic acids like DNA and RNA all contain nitrogen. So we have to be able to get nitrogen into our cells. And bacteria are the only living things that can actually convert nitrogen from the atmosphere into usable nitrogen in the form of nitrates and nitrites. So bacteria do that, and that way the nitrogen is available for all living things. Protista, algae, plants, animals, fungi can all use the nitrogen that has been converted from nitrogen gas in the atmosphere into nitrates or nitrites by bacteria. So then there is a video. I'm hoping it'll just play. Yeah, you just watch that. It doesn't have any narration in it, but this just shows you oscillatoria are also, just like the Anabina, they are filamentous, and they move by a gliding movement called oscillatoria movement. So go back to this. Then you have some of the cyanobacteria that grow in clusters. That would be the Aphanothece species and the Microcystis species. The nostoc, as you can tell over here, is filamentous. And then also the Anabina and the oscillatoria are filamentous because they grow in long filaments. So Anabina and oscillatoria and nostoc are filamentous, and then the ones that grow in clusters are the Aphanothece species and the Microcystis species. And so hopefully this will help you. Again, this is not due until Tuesday the 15th if you need that long to complete it.